everyone. My name is Jeppe Hallgren. I'm going to talk today a bit about how you sort of can turn machine learning models into more usable web tools and APIs, because that's really what matters when we talk about generating impact with machine learning. Typically, people do a lot of great work with the research, but never actually get it into the hands of the end users, and as a result, a lot of impact is lost. There will be sort of an interactive workshop element to this. You can find it on this link if you're interested. If not, don't worry, I'll go through it all in the end. So first, quickly about me. My background is actually in computer science. I studied in the UK before eventually transitioning into a master's degree here in Copenhagen in bioinformatics. So most of my work has been with machine learning. Did some work back in the days on advanced recurrent neural networks. Today, everyone uses LSTMs, but that's another story. And then I spent a lot of times on bioinformatics, specifically proteins. They've always been sort of fascinating to me because they're extremely complex molecules, and actually so complex that we can't really simulate the physics behind it. But that obviously opens up for a great opportunity to apply machine learning and get really interesting results. In doing so, I, for instance, wrote a machine learning framework called OpenProtein, specifically to make machine learning predictions from protein data. And I use that to do some transmembrane predictions and some other stuff as well. So it's on GitHub if you're interested. What this talk is about is what I spent the last 2 years doing. And it's specifically how do we as sort of bioinformaticians or machine learning researchers in general, how do we share our work, right? Because one thing is sort of taking your work and writing a paper about it. But most people can't really use your paper about that machine learning model to actually generate insights in their own work. So the impact is rather limited. And so the most important thing to take away for today is, well, maybe it's a small exaggeration to say this is the most important thing in machine learning, but I think at least that's the most overlooked thing is basically accessibility. How easy is it, at the end of the day, for your end user to take your model, your machine learning model, and actually apply it to their own data, right? Because if they can't do that, the research isn't really worth that much. Cool. So let's take a step back and talk about how do we sort of in general scientific world, how do we share results, right? And so the most typical thing is that a researcher will be in the lab and they will discover something of interest for the rest of the world. So they will write a scientific paper about it and they will publish it in some journal. And that tends to work pretty well, right? That's how sort of academia in general have been going for many, many years now. But I think it's very key here that, if you read about some interesting molecule in a paper, you can then use that directly as part of your own research. So the research output in science is quite consumable by the end user. I'm of course talking about general science here, so let's try to be a bit more specific. If you take a subject like computer science, which I studied, right? Typically, when you publish something in computer science, you will publish a paper with your findings, but you will also publish your code, right? Typically on a platform like GitHub or GitLab, you'll basically share your code directly with your peers. And that tends to work relatively well, right? Because basically the consumers of this research will also be computer scientists that know how to read code, but also to put code into use, right? So typically they can take whatever you've done and basically build on top of that, right? And that's very key here, right? That they don't just learn from your research, they can actually use your research directly on their own data. However, if we then jump to something like bioinformatics or more generally biological data science, we sort of have a problem, right? Because we can definitely still publish a paper, and we can also share the code. But our end user typically is someone who is a biologist or chemist who are not comfortable with running code directly from their own laptop, and that basically causes a huge accessibility problem. And at the end of the day, means a lot of the really cool machine learning algorithms that are being published in life sciences are never actually put to use, simply because the end user can't consume them directly. So what can we do about this? One of the most common solutions is to build what, if you're in bioinformatics, you've probably heard the term web server. It's not quite the same as if you come from computer science, but at least in bioinformatics, there's this term called a web server, which essentially is a website you can go to in any web browser, so it's very easy to access. you can just paste in your data, in this particular case on my screen, a protein, and press run. And essentially the website will then crunch the numbers and return you the result directly for you to consume. So this is very neat, right? Because now suddenly you don't need to know how to run a machine learning model or how to run C++ on your computer. You can basically consume this research directly through your web browser. However, creating web servers is not so simple, right? So if you imagine here on the right, essentially you have some researcher in the lab that want to consume your research. So you code, in this case, if DNA, but imagine it was your machine learning model. They need to do that through some server, right? So the minimum thing you have to do is basically figure out how to do server hosting, right? So your code needs to be accessible through the internet. Then you need to develop some kind of front end. Not to go too much into the details here, but for those who are familiar with web development, you probably need at least some degree of knowledge of HTML, CSS, JavaScript. It's a pretty steep learning curve for someone who just wants to publish their bioinformatics research. And finally, if you succeed with all of this, you still need to do some basic data processing, going from whatever the user enters into your website to what your machine learning model actually consumes. So there's probably some pre-processing and validation there. Finally, you typically develop your model on your own laptop, but now suddenly this is supposed to run on a server, maybe in the cloud. How do you need to make sure it runs exactly in the same way on that particular server? And finally, queuing, this is where most people sort of miss out on. Right, if you have multiple people at the same time using your server, it likely won't work unless you build some kind of queuing system that basically processes one request at a time. But all this work just so you can publish your research. And actually, as a consequence of this, most researchers don't do this, right? So they just create the paper, but never actually put it into use. Unfortunately, the story gets even worse, right? Because what about data security? Right, so what's happening when you're actually using one of these web servers is that you're on the left here, you're the researcher with some interesting data, and you now want to apply this machine learning algorithm on a remote web server. For this to work, you actually need to send your data over the internet to this server for the server to process it. This causes a lot of problems, right? Now suddenly, you're transmitting data over the internet to some third party you may or may not trust with your data, right? Imagine if this data was a proprietary molecule at your company, or it might be some GDPR data from the hospital. Can you just basically share this data over the internet? Maybe, maybe not. However, if you're in the industry, you almost certainly can't do this, right? And this actually means that most web servers are not accessible to people in the industry because of IT requirements. You just can't use some third party's web server. And that's really a shame. And that's sort of the point of this whole talk is, how do we create impact also in industry, right? Because, that's also a very important user group. So overall, too few web servers are created, and those that are created are typically not accessible by the end user because of data security and various numbers of other reasons. So most people just default to open sourcing their code on GitHub, and I really think that's great. I love open source. But unfortunately, for the people specifically in the life science industry, the users are typically not comfortable with consuming the research directly from that. And this is obviously a missed opportunity for people in academia. They could go more citations, but also the general sort of philosophical thought. We as humans can only really make progress if we're able to apply each other's research. So I think this is a very critical problem to solve. Cool. So what I spent the last 2 years on is basically trying to come up with some solution to this problem. Can we make it easier for people to share their research? Can we make it easy for them to build not only web servers, but also secure web servers, so we get around all these data security problems? And so what I built is this. You can think of it a bit like an editor. If you use Photoshop for photoshopping images, this is a tool for creating web servers. So on the left here, you have your source code. This would be a lot like something you have on GitHub. On the bottom left, you can sort of configure what inputs your users should be prompted for. If it's a server processing protein sequences, you would probably set up some sort of protein parser. And on the right is basically what the user will see. That is the web server where they can consume your research. I'll go much more into this. But I thought it could be fun to actually try this out in practice. So let's hypothetically say we wanted to create a web server for promoter classifications. So we want to build some kind of machine learning model specifically to predict if a DNA sequence is a promoter or not. And without going too much into the biology, on the left here, you have a diagram. Sorry, on the bottom, you have a diagram with a double-stranded DNA string. And on the bottom string, you have a gene to the right. And right to the left of the gene, upstream in the sequence, you have something called a promoter region. A promoter region is one of several factors that basically will cause the RNA polymerase to start transcribing the gene. So it's quite interesting for us because the promoter region is always at the beginning of a gene, right? So if we had a reliable machine learning model for essentially classifying if a DNA sequence was a promoter or not, we could take the full genome of a new sequence we found somewhere in nature and quite quickly basically find all the genes inside that genome because we could just look for the promoters, right? So if this was possible, it would be a really neat machine learning application, and lots of people could basically benefit from that right away. And the challenge here, of course, is both to build out the machine learning model, but also to deploy it in a secure way that's accessible for other people. I'll just talk for maybe 10 minutes about how this actually works from a technical point of view. But if you just want to get started and building the machine learning model, feel free to do that. The link is up there. Cool. So I think all the things about sort of building a web front end, etc. That's kind of annoying and it takes time, but it's doable. I think the really interesting thing is how do we actually get the data security? Because especially for the ones in industry, that's really what makes the difference between you generating impact or not generating impact. So I would like to define this term we call zero knowledge. It's essentially, can you use this service without leaking a single bit of information about your data? And it's quite simple. If you're leaking anything about your data, basically, how can you really trust it? So what we want to do is we have the data on the left here, then we have some network connection, and then we have the server on the right containing the machine learning model. So the first option, and that's probably the simplest one to understand, is what if we turn this error around? What if instead of sending the data to the server, what if we just send the algorithm to the data, right? So we do client-side computation. The immediate benefit of this is, of course, if you never move the data, it's perfect security because you do all the work client-side. We still want it to work in a web browser, though, right? Remember, we can't sacrifice accessibility, right? We can't rely on the user being able to run some Python script directly on the laptop. They're most likely on some corporate Windows installation where they can't just run code. So we don't want to sacrifice usability. So that's one option. The other option I'll also go through is what if there's some way we could encrypt the data before sending it to the web server, such that we didn't leak without leaking the decryption key to the web server, right? What if the web server was able to work directly on the data, on the encrypted data, without accessing any clear text? Then you would also achieve zero knowledge data security. Cool. But let's start with client-side computation. So the idea is basically, if we could execute the code directly in a web browser, then we have the best of both worlds, right? The data never leaves your own computer, but it's still very accessible. You just need a web browser, and most people in the world have that, even those without any big technical setup. If you're a web developer, you're probably familiar with JavaScript, which is the most used language on the web. It is in principle Turing complete, and that, without going into too many technical details, it basically means you can compute anything in JavaScript. It's not great, it's not going to be fast, and it probably won't run code such as Python, C++, or Rust, but it could work in principle, right? You could take any machine learning model in the world, and you could write in JavaScript. That would work. But how can we make this slightly better? Well, it turns out a lot of smart people behind the major web browsers, Chrome, Firefox, etc. They basically got together and designed a new standard for doing computation on the web. It's called WebAssembly. It's an instruction set, but you can think of it as sort of a very simple programming language that runs directly in any modern web browser. And it's been doing that for over 3 years now, 3 years now. So it's pretty stable at this point. Because it's a new bytecode level language, it's not emulated or simulated. It runs directly on your machine. It's almost as native speed, which is very nice when it's run fast. And it has quite a wide language support. So very interestingly, we can now run things like C++, Rust, Julia, Python directly in our web browser, which is very cool. So to try this out, for instance, I converted SAMtools, which is the most popular genomics tool, to WebAssembly. So what you see on the screen here is basically the command called SAMtools stat, running directly on a 350 megabytes BAM file, a genomics file, and producing the output. This runs directly in my web browser client side. So this 350 megabyte file, it never left my computer. Right? That's both nice for network bandwidth, but also really nice for data security. The speed is definitely slower, right? So in this thing, we're around 45% slower. But for most users, whether it takes 6 seconds or 10 seconds, it's not going to make a big difference to the day-to-day work. So overall, I think this is a very nice solution for the problem. That said, it's still very early days. Many libraries are not supported. I spent a long time getting, just the basic Python packages to work. It works now, though. That said, if you use something slightly more exotic, it probably won't work. Multi-threading is still very experimental. And finally, you have the problem that when you're building something with WebAssembly, you're probably building for many different browsers because it is a standard, but it's still early days. So I think it can be slightly different in each browser. And finally, I didn't have that room for that on this slide. But one of the major hurdles is also that you're running on the user's computer, right? And you don't really know what computer they run on. So even though it'll run, it can be a really old computer that runs really slowly, right? So your user might see different experiences depending on what laptop they have. So,
Jeppe?
and yes?
There's a question from the audience, from
Yes.
another Jeppe. Will it be able to run MATLAB code?
Yes, actually it will. I can show a demo of that afterwards, but actually we have a blog post for that. The trick is basically that MATLAB will compile to C++ and C++ will compile to MATLAB. But again, I can show you a demo afterwards if you're interested.
Thank you.
Cool. Yes. So the other option for doing zero knowledge computation is basically, if there was a way we could transform the input data before we send it over the network, such that it was unreadable by the web server, but while at the same time being able to do the same machine learning or computation on the data, right? Then we would still have zero knowledge security, right? Because we wouldn't leak any information to the web server. There are a few options for these quote unquote encryption systems here, because the first one is anonymization, and I think this is probably the most used one, even though it shouldn't be. Right. So the typical idea is if you just remove anything personal identifiable from the data, then it should be okay to send to a third party, right? Because they can't really do anything with it. Except in practice, many things, especially biology, are uniquely identifiable, right? So if you're working on a secret drug for the cure of some disease, right? You're probably the only one in the world working on that particular drug. Or if you are doing analysis of your own genome, right? You're very likely to be the only person in the world with that genome, right? So the data itself uniquely identifies you, and that makes anonymization extremely hard. And I think in many applications in life science, actually just not a feasible solution. Another really cool technology is called homomorphic encryption. And so the idea is, can we map our input data into another mathematical space where mathematical instructions still work? So we still do addition, subtraction, basically anything a computer program would do on data. But retaining the data inside this mathematical field where it's unreadable. Then basically we could do all the number crunching in the encrypted space. And then whenever we're done, just send back the encrypted space to the user, and the user could then decrypt it. This is a really, really neat technology. It's sort of called the holy grail of computer science, I think. And I spent many, many months trying to get it to work well. It does work for some cases, but the main problem is that it's really slow. It's at least 3 orders of magnitude slower than native computing. And I used to think this wasn't a problem, but I've learned the hard way that it actually is. It's not great if a machine learning model takes a couple of minutes to run and suddenly you're talking days. And again, remember, the most important thing is accessibility. So we can't sacrifice that. Then it just won't work. The final option is something called secure enclaves. It's a relatively new technology. It's very cool. So the idea is, imagine you had a special hardware chip. Think of it as sort of a black box. Actually, this is not hypothetical. Most CPUs today, you buy a modern CPU in the store, it will have a secure enclave inside it. But it's basically a special chip that can run something we call can prove to its user that it's running a particular piece of code, and that it only operates on encrypted memory. These are the two properties. Now, why is this very important? Well, if you could can sort of trust that a chip runs a particular set of code, and you have seen the code, and you know that it only works in encrypted memory, you can kind of guarantee that whatever data you send to that particular chip won't leave it, right? Because you've seen the code, you know exactly what the code does, and you know that whatever memory it accesses will always be encrypted. These are two extremely powerful properties. So how does it work in practice? Well, if your such a chip exists, and it does, it is in all modern computers, then basically what we can do is we could have the, so on the left here, again, I have the data, on the right, we have the web server with the secure enclave chip this time. What we can do is we can, inside this chip, we can generate an asymmetric key pair. We can send the public key back to the user. They can encrypt the data using the public key, send it back over the network. The hardware chip will then receive the encrypted data. It can decrypt it because it generated the asymmetric key pair inside itself. It can now just do compute on it, right? You can see the decrypted data. It can re-encrypt it and send it back to the user. Now, this is a lot of steps, and sorry if this is going too fast. But the basic idea is you achieve something like end-to-end encryption, right? Your data is never decrypted on its way to the chip. No one can access it inside the chip, and it's re-encrypted before it leaves the chip again and goes back to you, right? So not even the web server can see your data, right? Whoever is offering this web server can't even see your data. So in that way, you basically achieve zero knowledge data security. I hope this is sort of makes sense, but it's extremely powerful, right? Because it now means we get all the benefits, right? You have zero knowledge security. You can process it on a remote server, so you have the full cloud at your disposal, right? You can run 6 to 8 cores if that's what you want, and you no longer rely on the setup on the end user computer, right? So even if they have a really old laptop, it will still run exactly at the same speed as if you had a really fast laptop because it runs on the server. Let's try this in practice. So I'll now go to this. Actually, let me show you. I have prepared a. Oops, hang on. Just need to get this to work. I prepared this workshop at link.biolib.com/Odin. It's a Google Colab notebook. Maybe you're familiar with Google Colab already, but it's basically a Python notebook where you can quickly step through and see someone else's work. The first thing I'm going to do, maybe you already started on this, but basically the first thing I'll do is I'll copy it to my own drive. This is just so I can sort of make changes to it in case you want to say update the machine learning model. Cool. So the data set we're going to be working on today is a data set of promoter gene sequences. It's from the UCI machine learning repository, which is a great resource for data science data sets in general. And the task, I already went through it, but it's basically given a DNA sequence of 65, I think it's 67 bases, try to predict if this is a valid promoter or not. It's a quite simple task. It's just binary classification. So the first thing we'll do is download the dataset. I'll just start now. It may take a bit of time. It's not a particularly big data set, but sometimes the internet connection is not great. The data set itself basically consists of, I think, something like 120 sequences. I am going to use pandas here. Maybe you're not familiar with it, but it's a really nice Python library for manipulating data sets. So I'm going to read in my promoters.data file, and I'm going to change the column names to something more human readable. So then I basically get this back. So as you can see, we have 106 rows of data. We have a label column, which either using plus or minus says if this is a valid promoter. We have a sample column, which basically says where this particular promoter is from, what gene. And then we have the DNA sequence of that particular promoter, or in the case of the negative samples, non-promoters. You can read a lot more about the data set on a link above if you're interested. So the first thing we're going to do is, if you've done machine learning before, you know that machine learning doesn't work particularly well on letters. So the first thing we want to do is want to map this data to numbers instead. So I'm going to use, actually first I'm going to drop the sample column, right? We don't really want to be training on the sample because that's just the name of the promoter. So I'm only using the sequence and the label column. Then I'm going to take each base in the DNA string and map it to an integer, A to 0, T to 1, C to 2, and G to 3. And I'm going to apply that to all the sequences. And for each label, if it's a minus, I'm going to say that's 0. And if it's 1, sorry, and if it's plus, it's going to be a 1, so a positive sample. And then I'm using this function from sklearn to basically just split my data set into a training set and a test set, just so we can see roughly how well we're doing. And in this case, I'm basically printing out my training set. So as you can see, we have now 63 rows. We reserve some of them for testing, and we have 57 columns. Basically, for each base, we now have 0, 1, or 2, or 3, or 4, marking each of the 4 bases in DNA. So now we're basically ready to do the machine learning part. I'm going to use a library called sklearn, specifically k-nearest neighbor from sklearn, which is a relatively nice model. It basically just takes the input data, see if we have some similar ones, and then it tries to classify the input sample according to those similar data samples. So I created this model, I trained my model, and then I tried to evaluate my model on the test set. In this case, you can see we get an accuracy of 69.7%, which is pretty good. Random would be 50. So you always have better than 50, otherwise you're not doing so well. Feel free to try to play around with this. We could change the number of neighbors to 5, see if that improves accuracy. It actually does. Now it says 74.4. So basically, feel free to play around, and you maybe even try to replacing the k-nearest neighbors with a decision tree or something else. Cool. So now to the more interesting part. So now we basically have done all the hard work of cleaning our data, training our model, and now we basically want to generate some impact with this model. So the first thing we need to do is basically export the model. And in this case, I'm using a library called BioLib, which basically can save my model to disk. So I'll just run that. In Google Colab, you have this small icon out here where you can see your files. So if you open that, you should now have a model.joblib file. This is essentially our trained machine learning model. I have then written some code. So this is probably what you might have done in the past if you tried doing this yourself. It's essentially some code to do inference, right? So we're going to have some code that takes in the user's data. In this case, I've just hard-coded a DNA sequence of 57 bases. We then have to apply the same mapping to the data as we did when we did the training. So mapping DA, TG, and Cs to integers. Then load basically the model in from disk, and then make the prediction using the model on the data, and then basically print out whether this was a promoter or not. So if you run this, this model basically predicts that this sequence is indeed a promoter. Okay. Very good. So this is what we were doing in the past. And you'll find many examples of people then sharing this code on GitHub and saying, hey, you can run this model like this. That's, of course, only good if the user knows how to run Python. So now let's try building a secure web server. So in the interest of time, I have sort of created a base sklearn application set up with all the right dependencies. So if you go onto the link and just fork this, you should essentially get a copy of it already set up. So then everything should already be in order. So this is what a default web server looks like. It's not particularly interesting. It basically just takes in a name, a variable, and it prints hi there, and then the name. You could change this to hello, and then save. So now basically we already have a web server. So this is a link that could be shared with anyone. So your peers in academia or your colleagues in industry, and they right away could go in and actually run this code. They don't need anything else other than a web browser. And what will now happen for you behind the scenes is basically we'll do the secure enclave encryption as I told about. So basically we'll encrypt input data. In this case, just my name, Jeppe, not particularly interesting. It will then send it encrypted directly to the enclave, which will decrypt it, perform the computation, re-encrypt it, send it back to me, and then decrypt it and show it in my browser. So all of this happens relatively quickly. But the interesting thing is now that just anyone in the world with this link can now go in and run this code. Of course, printing my name is not particularly interesting. So let's try to do something slightly more complicated. So now let's say we want to change this to, instead of a name, accepting in a DNA sequence. This is, of course, for our promoter prediction. So I'll scroll down and I'll change the input to a sequence. Text input is fine in this particular case. And we can ask the user for a DNA sequence instead. And maybe we can specify that it should be 57 bases. Then we specify a default value. Remember, the most important thing is accessibility, accessibility, accessibility. There's always a very good idea to show some example data to your user. It's just a lot easier for them to then map that example data to their own data and understand what changes they need to make. So let's go in and I'll just use the sequence I used in here. Hopefully we should get the same result. So now if I save it now, basically the input has now been changed from asking for my name to instead asking for my DNA sequence of 50% bases, and it has some default value. I could then go and override if I wanted to. Cool. Of course, this still just prints, actually it prints the wrong thing here. So I'll just change it to reading a sequence instead and print the sequence. Yes, so now let's try to do something slightly more interesting than just printing the sequence. Let's try to actually apply the machine learning model we trained on Google Colab on this particular sequence. So I'll go back to Google Colab. I'll actually just copy the same inference code I had here and paste it here. We no longer need to print it, so I'll remove that. Of course, we shouldn't hard code the user's data to this sequence. That would be not so exciting. So instead, I'll just change that to be the input from the user. Great, except not so fast. So this is the inference code, and it runs fine. But if you remember, it's actually loading this model from disk. So of course, we need to supply this model as well to the application. So if you go back to my notebook here, remember the model can be found out here on the right, and you can actually click here and say download. And that will now download the file to your desktop. So now I should be able to go in here and upload this particular model I've trained. It's here. I'll just rename it to not have, oops, to not have this brackets 2. Great. And actually, it looks like I placed it inside the source folder. So I'll just add source here to this path. Okay. So assuming I did everything correctly, I now have this DNA promoter prediction web server. Let's try giving it a sequence. Just give it a sequence. And it will actually now, again, do the same encryption I spoke about previously, then map the input sequence I just gave it to numbers using that dictionary I use for translating each base into a number. And then we'll run the sklearn model on the data, get the result, re-encrypt it, and send it back to me as the user. So in this case, we can see that it actually predicted this particular sequence is a promoter. And then we're basically done, right? So hopefully this was not too difficult. We essentially managed to go from a model we had trained in Google Colab, or if you use machine learning, you probably already have your own machine learning setup, to actually having a web server we could share with anyone, and they right away could start generating insights on their own data. Of course, a really nice property and important property as well is maintainability. Right, so a lot of people, when they publish an academic paper, it's very hard to make corrections to that paper. It's possible, but it's not great. We want that to be easy for these web servers, right? Because people are actually going to be running it on their own data. So one thing you could do, oops, sorry, it's very easy for you. So let's say it's kind of annoying how I was printing that long sequence again. That was my input. So maybe I just want to print a sequence instead of actually printing the full sequence. Now, right away, it has been updated. So if you look out here, you can actually see the different versions I have released during the course of this demo. This is very critical because we're obviously talking about science here, right? And reproducibility is almost the most important thing. And that's something you typically lack with traditional web servers, actually. But it's very important that if you put in your paper, hey, I went here and I used exactly this web server, someone else coming along 2 years later and running the same thing, they should get exactly the same result. Another really nice thing about these enclaves is because we're now nicely wrapped it inside this box. It's quite easy to add other interfaces to it. Again, most users will probably prefer some web interface, but if you have a technical user that would rather, want to interact with this application through an API, they can do it from Python or directly from the terminal. The point is just that, we want to make this accessible to the whole range of users, and especially in industries like life science, the technical abilities of each user can vary a lot. So it's very important that you have multiple interfaces for your work so people can actually apply it. Cool. Yes, I think that's basically it. Again, feel free after this workshop also to play around with it yourself. You can do more interesting things like maybe prompting the user for a file instead of a sequence. A lot of times people have these files containing lots of different sequences. So it's quite easy for you to just swap that in with something else. Yes, cool. So going back to my slides here, I think that's basically all I had. What's next? More building. as you learn quickly, bioinformatics have a lot of different frameworks and languages, so it takes really time to get good compatibility with everything. I think this project has gone a long way, but it can also always be better. So I'd love to hear from you if you try it out and have any comments. Feel free to join on Slack. It's probably the easiest way to reach me. We're also doing a lot of hackathons, both to sort of build out this project, but also to just build machine learning models in general that can be used by a wide audience. So if you're interested in joining those or maybe even co-organizing some hackathons, also please feel free to drop me an email. Cool. That's all.